Fungal zoonoses or mycosis. Fungal zoonoses or mycosis. Fungi are eukaryotic cell walls similar in structure to plant but vary in chemical composition. Chitin, glucose, and mannan. Cellus body unicellular yeast or multicellular molds. Mycelium molds. Loosely organized mass of filamentous hyphae. The characteristics of fungi: body form unicellular yeast, multicellular mold, filamentous, tube-like stems called hypha, similar or hypipular. Mycelium aggregate of hyphae, sclerotium, hardened mass of mycelium that generally serve as an overwintering stage. Multicellular such as mycelia, mycelial core, rhizomorph, and fruit body, mushroom. Fruiting body, both are composed of hyphae, mycelium, hypa, septate versus cosinocytic, non-septate. The characteristics of fungi: heterotrophy, other food. Saprophytes or saprops feed on dead tissue or organic waste decomposure. Symbiont, mutually beneficial relationship between a fungus and another organism. Parasite, feeding on living tissue of a host. Parasites that cause disease are called pathogen. Heterotrophic by absorption, fungi get carbon from organic source. Hyphal tip release enzymatic breakdown of substrate, produce diffuse back into hyphae. Nucleus hang back and direct, produce diffusion back into hyphae and is used. Hyphal growth from the tip. Mycelium, extensive feeding web of hyphae. Mycelia are the ecologically active body of fungi. This wall is rigid, only the tip wall is flaccid and stretches. Modification of hyphae. Arthrobotrys oligospora. Arthrobotrys oligospora. Nematode tapping fungus. Hyphal growth from spore. Germinative spore. Mycelium. Mycelia have a huggy surface area. Mycotic zoonoses. Superficial mycosis, outermost layer of the skin and hair. Tinea, viscid color. Pideria, hot track. White pedra. Cutaneous mycosis, cutaneous tissue epidermis, mycosporum, trichophyton, ringworm of lesions. Subcutaneous mycosis, subcutaneous Sporidium severi, sporothrix, sporotrichum, Schanke. Rose gardener disease. Systemic mycosis, lungs, aspergillosis, blastomycosis, candidiasis, cryptococcosis, histoplasmosis, zygomycosis. Dermatophyte or tinea or ringworm. Dermatom means skin, phyton means plant that grow on the skin. Trichophyton, verrucosum, microsporum, epidermophyton, flocosum. Trichophyton, mentagophytes var interdigitalis, microsporum nanum, epidermophyton, stockdale. Trichophyton, tonsurensis, microsporum, gypsum. Trichophyton equinum, microsporum, Trichophyton tonsuren. Arthrophilic, zoophilic, geophilic. Epidermophyton, flocosum, M. canis, M. gypsum, tuberosum, M. nenum, M. odinini, M. distortum, M. cookie. Microsporum, hyphae produce chain of spore-forming sheath around the hair shaft. Ecothrix, Trichophyton, hyphae produce spore within the hair shaft. Endothrix, hair become weakened, typically break easily at the follicle opening. Tinea capitis, infection in scalp and hair. Trichophyton or Microsporum. Tinea barbae, infection in beard area. Tinea corporis, infection in the trunk. Tinea crucis, jock itch, infection in groin and inguinal region. Tinea menum, infection in hand. Tinea pedis, athlete foot, infection in foot. Laboratory diagnosis: simple skin scraping, nail scraping, hair plucking. Collection of sample skin from the margin of the lesion with the scalpel nail. Deeper part is collected and superficial part is discarded. Hair plucked by fine forceps. Wood's lamp test. Ectothrix of Microsporum species import a greenish to silvery fluorescence when examined under wool's light. Infectious organisms glow under wool's lamp illumination. Microscopic examination. KOH, preparation of skin or nail branching hyphae or chain of arthroconidea are seen. Utensil: unstained microscopic KOH prepared of scraping for a ringworm showing arthropods, a sexual spore. Multicellular microconidia of M. canis, elongated microconidia of T. tonsurans, club-shaped microconidia with thin and smooth wall arise in small cluster of E. fluorocosm. KOH preparation of hair. Ectothrix and endothrix are seen. Ectothrix, arthrospore outside hair shaft. Endothrix, arthropore inside hair shaft. Culture: incubation period 1 to 3 weeks. Incubation temperature 25 degrees Celsius. Media use: Shubbard's dextrose-agar media, dermatophyte test media, Shubbard's dextrose-agar plus cyclohexamide plus chloramphenicol plus bisphenol red, malt agar. Colony morphology. Microsporum white coaty surface with deep yellow foam ribs. T. tonsurans flat powdery velvety colony. Epidermophyton flat velvety with a tan to olive green tinge. PCR species specific identification. Systemic mycotic zoonoses. Aspergillosis. Aspergillus is a ubiquitous gene of mold that is commonly found in soil and decaying vegetation. Aspergillus fumigatus, most commonly found species 90%. Other species that can cause human disease include Aspergillus flavus, Aspergillus terras, and Aspergillus niger. Aspergillosis is not contagious. Aspergillus cannot be transmitted from person to person. In outdoor environment, soil decomposed heap, damp grain inside building, damp insulation, fireproofing material, bedding behind shop, in the corner of damp room, dust, air conditioning system. Farmer lung disease, hay, animal feed. Invasive pulmonary aspergillosis, IPA, fever, pleuritic chest pain, Pneumothorax, coughing of blood, cough and shortness of breath. Treatment: voriconazole is recommended as first-line therapy of IPA. Voriconazole is recommended as first-line therapy for IPA, chronic pulmonary aspergillosis, CPA, allergic bronchopulmonary aspergillosis, Aspergillus rhinosinusitis, invasive aspergillosis, voriconazole, amphotericin B, isavuconazole, posaconazole, echinocandins, chronic pulmonary aspergillosis, itraconazole, voriconazole, posaconazole, isavuconazole, amphotericin B, echinocandins, allergic bronchopulmonary aspergillosis, corticosteroids, itraconazole, voriconazole, nebulized amphotericin B, aspergillus bronchitis, itraconazole, voriconazole, nebulized amphotericin B. Other important fungi are included in the group: Histoplasma capsulatum, Blastomyces dermatitis, Paracoccidioides, Baracellensis, Coccidioides imitis, Cryptococcus neoformans. Four of the pathogens are dimorphic: H. capsulatum, B. dermatitis, P. Baracellensis, C. imititis. They grow as filamentous mold as spore and in culture at 25 degrees Celsius. When they infect human or are cultured at 37 degrees Celsius, they transfer to a unicellular morphology. Histoplasmosis, also known as Darling disease. Histoplasmosis, known as Darling disease, acute pulmonary flu-like, chronic pulmonary or disseminated. If untreated, the symptom may be purulent or bloody. All are endemic to certain geographical area where they can usually be found in the soil. Blastomycosis, Blastomyces dermatitis, Blastomyces dermatitis, Gilchrist disease. Amphotericin B remain is the drug of choice, DOC, although it is very toxic and must be administered intravenously for several weeks. Ketoconazole is also being used in mild cases. Coccidioides imititis, valley fever or California disease. Desert soil, pottery, archaeological middens, cotton and rodent burrow, all herbaceous. C. imitis is a dimorphic fungus with two life cycles. The organisms follow the saprophytic cycle in the soil and the parasitic cycle in man or animal. Cryptococcus neoformans, a capsulated cyst, a twist, a sporadic disease in the past, most common infection in AIDS patient. Life cycle of neoformans. Candidiasis. Candida albicans is an opportunistic fungal pathogen that is responsible for candidiasis in human host. Candida albicans grow in several different morphological forms, ranging from unicellular budding yeast to true hyphae with parallel side wall. Candida albicans is a unicellular oval-shaped diploid fungus, a form of yeast, typically Candida albicans live a harmless commensal in the gastrointestinal and genitourinary tract and are found in over 70% of the population. Overgrowth of these organisms, however, will lead to disease. Candidiasis is a fungal infection that can affect areas such as the skin, genital, throat, mouth, blood. Thanks to all of my students to listen attentively.